today we are going to see, just analyze the questions and then discuss all the questions which are from unit number 5, 12 and then 13 from our recent question paper, that is June 2012. Okay, before going to that one, in the development biology class I have taken about all the different cell fate mechanism, and then I found this flow chart. It will be more interactive, and then it was like very crystal clear. That will be helpful for you when you are going for the exam. So we know that the zygote will be formed by the fusion of your sperm and ovum, and then the morula will be formed. That is the in between intermediate stage of blastocyst. So once the blastocyst is formed, it will have the inner cellma, that is nothing but your pluripotent stem cell, whose fate will be determined. Okay, so you guys know that this in blastocyst will be implanted into the uterus, so where the gastrulation will take place. The major event during gastrulation is the formation of 3 different layers. One is ectoderm, mesoderm and then endoderm. So if you see that ectoderm are the cells involved in the formation of eyelids, auditory vesicles, so that you can see that ear, eyes, and then you can go for outer epithelium of the body, the entire skin cells, and the neural tube, all your nerve cells, neural crest, there is nothing but your brain as well as your all your sensory organs. All of them are product of your ectoderm. It is straightforward. Remember this some way or other. I am telling the most easiest way. The next thing, the mesoderm, if you see that all of them are dorsal, paraxial, all of your skeletal muscles. And then intermediate, the kidney, uterus, vagina, oviduct, uterus, all of the product of your mesoderm. The lateral mesoderm, there is nothing but you can go for like the different layers in the mesoderm itself, that will produce all your bloods, gonads, and then flora, and then all your in between cells, and then your endoderm, that will produce your entire digestive system, and then it produces also your inner middle ear, thymus, all those things. Please go through this flow chart. They are asking just like that. It is very easy, straightforward question. You can crack that any type of questions based on this self-fate math. That is the reason I want to share this one with you guys. The next one will go to the question. I will do that today. Okay, it will be uploaded today, like today evening I'll upload it. And then in this you guys are asking about your differentiation and then pattern formation. In that another important factor is competence. I have seen a question based on this. Competence is nothing but ability to respond to a specific inductive signal. If you see that they asked for the definition in the first part of your unit number 5, first unit definition. Competitive is the ability of the cells to respond to a specific signals. So actually if you see that those competence, I am saying that this is related to your self-fate determination, right? So this competence cannot be acquired just by passing. It is not predetermined, which is not like already it is there. It will be acquired by condition. What is meant by conditional? If that particular condition required, then only that signal will be happening. Okay, so the competence are acquired only by condition. If you see that, for example, if you take that in developing a chick and then mammalian eyes, the gene, very important gene, PAX6 protein, appear to be very important in making ectoderm competent to responsive for the inductive signal from the optic vehicle. Okay, the thing is, like, this is the protein which act as a messenger. This will go and tell to the cells, ectoderm cells, hey, this is, you are going to be converted into your eyes. So please respond to the signal. Got it? If you are not, if you mutate that signal, what will happen? There will not be any production of your eyes or any ears, no sensory organ. If you mutate this PAX6, there will not be any ear or eyes. And then this actually, if you see that this PARC6 expression is seen in only the head ectoderm, which can respond to the optical vesicle by forming lens, which is not seen in any other region of the surface ectoderm. Okay, so it will be like present only that particular. If the PARC6 is mutated, that person is not going to have any ear or vision, or he cannot hear. So got it? So the person like some like deaf and dumb people, because they may be mutated with the PARC6 genes, because they have the cells for the production of that thing, but they are not able to respond because the PARC6 is totally mutated. And then moreover, this PARC6 is a very, very important competent factor. And then if you see that if the mutated, it doesn't, just see the picture. So this is your normal mice. It has your lens pit, so it will have your vesicle, all your signal is ectoderm thing. So if there, the signal is there, so that it can produce your nose and then the eyes. Whereas if it is not there, there is no any cells, the PARC cells are mutated, the baby is not going have any nose or eyes. So the PARC6 cells are very, very involved in the signaling process of for making your sensory organs. Understood? Clear? So this is your question number 94 in your part C. The functionality of a PARC6 gene in the formation of optic and nasal structure can be attributed by the following: A, B, C, D. Which one you feel like that? Online students, what is the answer for this? That's very fast. I got two answers from like that. What happened to others? A, Gurkamal, I think you didn't see the other options A, B, C, D, right? Is it confusing? How? C option is not there. Okay, then what is the answer then? B and C. Just now I explained, Ruby. C only. Let us discuss. The PARC-C makes a vesicle competent and then allows lens formation. Actually, it signals, it is not the one. The optical vesicle can induce any part of the ectoderm to form the nasal optical structure due to the presence of PARC-6. There was one thing I said, that it is going to be present only at particular position, not even on the surface of the head, right? So this, and then the PARC-6 renders the head ectoderm competent to receive signals from the optical. Yes, this is a refined version of A, I can accept it. Apart from optic vesicle, the head ectoderm may not be induced by BMP. These are all not at all a protein which are involved. The answer is the D only, C only, the straightforward answer. Any question? Yes, yes, yes. If the C is not the option, so we can go for your A. That is the most appropriate. The thing is, like, they will play with the words. So if you are not able to see, the thing is like by seeing A, don't check A. C. Okay guys, we'll go to the next one. Capsation. What is capsitation? Mm-hmm. So actually, if you see that the sperm cannot fertilize oocyte, that is, if you take any sperm directly from your testis, and then if you take a ovum from a female and then keep it in the test tube, if you add that sperm, do you think that the sperm will go and fertilize? It will not, right? It requires a specific environment for the fertilization. So that from this we can say that the sperm which is present in the testis has the capacity only to swim, not to fertilize a ovum. So this process will take place in the fallopian tube, that is a part of your oviduct or ovary. Mostly this process is around like 5 to 7 hours it will take, and then if you see that the agrosomal reaction cannot takes place unless this capsitation is not done to the sperm. Okay, that is in the videos and all we must have seen that. So we know the secret, right? So they'll give the hormone. So that is the reason mostly the test tube babies will end up with two twins babies, because we cannot control it. We have known everything till the blastocyst formation. So that is the reason we can put all the in vitro fertilization. After that we don't know. If we know all the proteins involved in the gastrulation, we can produce the baby outside. We don't want any mother, right? Definitely. There are so many people with so many problems, right? So we have a very good expertise as well as the very good knowledge till the blastocyst formation. Okay. So the capsation, so the process involved in the capsation, if you see that during the capsation, the process which is involved in the fallopian tube, what it does now, it destabilizes the agrosomal sperm head membrane, allowing greater binding of sperm and oocyte. The word is, it is going to do the destabilization. What is destabilization? That is the change which facilitates removal of any steroids, especially cholesterol and the non-covalently bound glycoproteins, which is from your testis. So all those things is there now. It will be unnecessary interference, so that the sperm will not go and bind directly to the ovum. Okay, so this removal of this glycoprotein and then cholesterol, which will result in the fluid membrane with an increasing permeability of your calcium 2+, so that what will happen? It will hyperactivates your sperm movement. Okay, so this influx of calcium 2+, which will increase your cyclic AMP and then increase mobility, that we have seen, that we know this. So this is the molecular event in very detailed manner. So next, the motility increases. So this hyperactivity coincide with the capsation and it results in the increased calcium 2+ levels, and then in which here the tripeptide, that is fertilization promoting peptide, produced in the male, which is very essential for your capsation. That is, if you see that the sperm which will be in the fluid, this fertilization promoting peptide will be present in that fluid. Okay, that will be activated, and then it comes in contact with spermatozoa only if it is removed. Initially it will be present in the liquid portion of your sperm ejaculation. Once this cholesterol and then glycoproteins are removed, so that this fertilization promoting peptide will find a place for go on binding to the sperm, which will increase the cyclic AMP activity, which in turn it will increase your calcium 2+ ion influx, which will induce the hyperactivation of the sperm. So this leads to the capacity of the sperm to go and bind with your ovum. This is your capsitation process. This FPP, it is not going to increase your calcium 2 plus. Once the calcium 2 plus ions are increased, this FPP go on bind. It will increase the hyperactivity. It is other way around. Okay. So now the questions. Online students. I got only 3 answers. Sangamitra, Anuja and then Preeti. Only 3 people answered. Oh, even Sangamitra didn't answer. Only Anuja and Preeti answered. What happened to others? I need answers from everyone. Okay. Okay, 1 or 2 people are there. Okay. This is your 95. Can you guys can read it, right? If you want, you can switch off the light. That will. Anuja, Preeti, what is the answer? Sangamitra. Preeti, what is the answer? What is the answer? So here they removed the sperm from the epidermis. This is the place where your mature sperm will be stored. So directly it is taking a sperm from the testis, and then what they did now from the male mouse, they added into the dish containing appropriate media and then oocyte. So they collected the male female oocyte from the mouse, and then as well as your male sperm was added. They are not able to find any fertilization. So the question you asked, in vitro fertilization, what they will do in vitro fertilization. So this must be the first step, right? They must have added the sperm and then ovum. No, nothing must have happened. But the sperm from the epidermis were directly placed in the uterus of the ovulated female, and then that mouse became pregnant. So this observation suggests that we need the capsation should be happened, right? Right? Once it travels, it doesn't attain. If you see that in a single ejaculation in man, it takes almost 300 million sperms will be entering the reproductive tract, and then it has to go through so many obstacles like your villi and then acidic condition in the vagina, and then plus your immune cells. It has to cross all those obstacles, and then it will reach the fallopian tube, and it reaches the fallopian tube, the number of sperms will be 1,000. This is an appropriate calculation. Just think about from 300 million to 1,000. If the nature that one cell can go, another man can produce only one sperm and then send it out, right? Or it has to produce 300. That too, if you see that, I think I have told that one to produce one sperm, it takes 64 days. Just think about how much energy is required for that. It is not a overnight process. Okay, we know that there are like so many obstacles. In that, among that 1,000, all the fallopian tube, all of them will be activated. All of them will undergo capsation. Among that capsated 1,000 sperms, only like 10 or 15 will reach the ovum. Among that, only 1 or 2 sperm will go and then just cross your corona radiata. It has to cross your zona pellucida, and then it has to hit your plasma membrane. In that, it has to go and bind your zona pellucida 3, and then it will go and attach to your zona pellucida protein 2, and then that will induce your hydrolytic activity of your agrosome. How precise it is. It is not like that all the 10 can go and fuse. Only the sperm which goes everything, which crosses all the obstacles, which follows the rules properly, and then that sperm will go and fuse the womb. So all of us are born like that only, right? So like that we have one almost like 300 million people, right? So in this answer, the most appropriate one is option number 4. You can get confused between 2 and 4 because the oocyte secretes some biochemicals and factors which helps sperm to fertilize. Definitely the oocyte is not secreting anything, so that just we can get crossed to that. So the content of the female reproductive tract interact with the sperm and then activate it is for your fertilization. That is the right option. Everyone, Preeti, Ruby, Sangamitra. Okay, next one. Next we'll go to the, I want to refresh about the amount of yolk. What are the different types of eggs classified based on the amount of yolk? Can you? Isoleucital, centroleucital, mesoleucital, teloleucital, and then aleucital. If you see that, this is your isoleucital, where very little yolk and then evenly distributed. Example is your Sation. And the next one is your mesoleucital, moderate amount of yolk concentrated at the vegetal pole. Always the yolk will be only present in the vegetal pole, not in the animal pole. Aleucital, very little or none. Example, mammals. And then your centroleucital, which is centrally present with a lot of yolk. Example, insects. The last, the teloleucital, lot of yolk at the vegetal pole, and then examples are birds. So these are the different types of yolk. So this yolk content is very, very important in the pattern of cleavage the cell is undergoing, right? The yolk content, if the centroleucital, it will undergo only the outermost surface alone will be going your cleavage. Okay, the yolk content plays a major role in deciding the type of cleavage it is undergoing. Okay, the type of cleavage. So the type of cleavage, basically it can be classified as holoblastic and then meroblastic. What is holoblastic? Holoblastic is a complete cleavage. Just cut the fruit into 2 pieces. Just the complete cleavage. Okay, the meroblastic, it is a incomplete cleavage. So the complete cleavage will take place in very, very important. Remember this, isoleucital and then mesoleucital. Whereas incomplete cleavage will take place in teloleucital and then centroleucital. Yeah, complete cleavage, yes. Okay, so you can put like complete cleavage, iso, meso, A. Then we can put AIM. AIM goes for your complete cleavage. Okay, so whereas your incomplete cleavage, the rest of the 2, TC. Transfer certificate, remember that one. The transfer certificate, if somebody gets, the course is incomplete. Remember this one, because more number of questions straightforward. Even the part C, you are getting like this. Which type of cell is undergoing type? Okay, so another way is, if you want to complete any course, you want AIM, right? AIM. A stands for aleucital, isoleucital, mesoleucital. That is your complete cleavage. If you get any TC, that is your teloleucital and centroleucital, incomplete course. Remember this one. Okay, easiest way you guys can remember this. The rest of the examples somehow you can manage. I'll show the option. Online students, you can get it right. Not able to hear. Okay, I think just now you entered. Maybe a little bit time it will take. Okay, so the examples for holoblastic, complete cleavage, and then under isoleucital, these are the examples for isoleucital. Don't worry about a radial, spiral, bilateral, rotational. Don't worry about it. Example for isoleucithal are echinoderms, amphimixes, annelids, mollusks, flatform, tunicates, mammals, and nematodes. Remember this. And then if you see the next one, the mesoleucithal eggs, are birds and amphibians. And then you are meroblastic. If you see that, it's a teloleucithal, TC. T stands for your cephalopods, fish, reptile, birds. And then centroleucithal, most of the insects. To be more specific, let us remember this one. There are different type of eggs, and then yolk content, and then type of cleavage, whether it's meroblastic or holoblastic. The result of is a blastocyst or blastoderm. Some of the examples which we can remember. Just go through this one. A type of cleavage, superficial. Okay. This is your question number 97 in your June 2012. Let us see who is going to crack this one. Gurukamal, can you hear me now? How? Explain. Did the AIM and TC help? Anuja, what happened to Preeti? What happened to Gurukamal? And then Sangamitra? So TC will not be a complete one. So gone. Right? AIM, I is there, definitely not a incomplete. So which is the leftover? The answer is that's right. Very simple, right? You can remember this one, AIM and TC. Okay. So next we'll see about our plant body. From 6th standard we are bored about this, right? Plant has leaf, stem, root. But there are questions in CSIR. So if we are seeing it that one for more than like 10 years, we should be in the good position to crack it, right? If we didn't crack that one, it's a big insult to us. That is what I feel. What do you guys feel? Where were you? I cannot repeat. Okay, so the plant. This is like your stem, leaf, and then root are involved in the absorption of nutrients, and then this plant's leaves are involved in the photosynthesis. And then here the thing is like a little bit mature question is how the plant grows. So there will be always one small analogy question will be there. Okay, it will be most analytical question. Okay, somebody like when the plant tree is like 3 feet height. Okay, somebody nailed iron piece at the 2 feet. After 15 years, the plant grow almost like 15 feet long. At what height this nail will be now? Can you imagine what I'm saying?
Yes, sir.
What is the answer for that?
After how many years?
15 years. It was 15 feet. What is the answer, Sadiq? Huh? It will be at that place only. Because? Why? Yeah, plant will be growing from the top. Wherever you hit, the nail is going to be there only. It is not going to grow. So the science behind is the plant grows based on apical meristem. Always Benazir is like that. Okay. Okay, the apical meristem is the one which will start growing, which will start adding the number of cells, so that always plant grow on the one side. It is not like in all the axes, x-axis, y-axis, z-axis. In the 3D form, it will never grow. It will grow only in one axis, and then these are the lateral growth, right? This also will have a apical meristem, and this is also. So the growth of the plant 100% required is your apical meristem. So let us see that. So meristem. So meristem is nothing but is a clump of small cells with a dense cytoplasm and then proportionately very large nuclei. It elongates your both your root and shoot. In some species, lateral meristem produce an increase in the that is nothing but branching. Some plants will have a lot of branching, so that is because of a lateral meristem. So apical meristem, which is always located on the tip of the plant as well as in the root. These are the tissues that results in the growth of your primary and then secondary, and then always the root apical meristem will be always protected by your root cap, because there should not be any damage to the root cap by the microorganism or by any soil pathogens. Okay, so that lateral meristem, if you see that this is a root cap, this is a apical meristem in the root, and then most in the lateral meristem, most trees, shrubs and some herbs have active meristem. Okay, it increases the non-woody plant, mostly the secondary growth. Woody plant, the cambium and then vascular cambium, these are the place where you all have a lateral. So it has only two type of meristem. One is apical, other one is lateral. Apical, it always moves on the straight, it will be in the leaf, in the tip, and the root in the tip of the root. Okay, and the lateral in the side, the branches. So understood how easy it is, right? Let us see now. This is your question number 96. How many questions till now in development biology? How difficult it is, all of them. I will tell that I will compare these questions like, what is your name? No mistake should be there in that. Online students? Somebody say someone answer. I think everybody, we should have an option here to close all your ears. How come? Define. How come the D option is wrong? Huh? How come the D option is wrong? So A is gone. What about B? Yes. Shoot is what? Your stem, axillary meristem. What is axillary meristem here? Side branches. It develops from your shoot apical meristem during differentiation of leaf primordia. Definitely the petioles, all those things. That is how it has to be, right? How come the branch can become out of the plant? It has to a deviation from your apical meristem. Lateral root develop not from epidermal cells. It is from your lateral roots or what? It should shoot. See, the thing is simple word. How the branches are forming. I am saying that the plants are growing only because of apical meristem, which is always on the tip. When the branch is coming out means what? The apical meristem should also present the branch, right? The branch is also growing straight from the tip. Understood or not? So that is what this is a mechanism how the branches are formed. And then root cap is made up of dead cells. If it dead cells, it will be removed by a bacteria very fast. And then the lateral meristem and the cylindrical meristem found in root and then shoot result in secondary growth. To be more specific here, cylindrical meristem. Apical meristem will be on the top, The side one is called your cylindrical meristrum. So the option is your, yes, Preeti.
So then lateral roots develop from where there is not from epidermal cells.
Lateral roots? Lateral roots are developed not from the epidermal cell. Definitely it should be from your apical meristem. The root which is present there, right? It is just like a branching of your root. Understood? It is not from the epidermal cells. With this information you can crack it. The answer is B and D, straightforward. So little bit analysis, and then thing is like one more thing, how to answer this thing now. If you don't know the option, don't try to justify that. If the first A option is wrong, all of them are wrong. Okay. And then one more thing, always the person who's setting a question, this is a very, very crude way of going to the answer. Okay, this is not anywhere, don't ask me any reference for this. This is my way of cracking. If I set a question, I will not consider any person who don't know the answer. Right? That is how I'll set a question. I don't want to set a question paper who is not going to answer it. I want to set a question who knows the chapter very good. Okay, always see for an option which subdivision A, B, C, D is more repeated. So that is right one. By seeing that, I can remove one option. Here the D is more repeated. It is present in 2, 3 and then 4. So definitely D is right. Yeah, that is what confusion. Now if you know this idea, definitely you will opt for 2, 3. So I want to confuse only with the person who is having half knowledge and full knowledge. I cannot touch a person who is having fully confident, but I can play with a person who is in between, cat on the wall, this side or other side, this or other side. So that if you are like this now, so mostly that is how I will crack. The one I will cross. So I'll end up with between 2, 3 and 4. I can directly cross 4 because this is wrong. So I'll be between 2 and 3. So if lateral roots develop from epidermal cells, from the outermost, epidermis from epidermis, I don't have any idea. Epidermis is skin. Do you think that hair in my body, the air is coming from the epidermis? No, that is a dead cells. How come that will produce one more thing? So I can crack it, throw it off. Without having any knowledge about my plant biology, I can go with B. This is called your part C analysis. Make sense or not? You cannot be a boss in almost everything, but you can use your experience. Got it? If you want to go to any other if you want to go to Chennai, you have never been there. Do you think that you need a big classes, 15-day course how to be in Chennai? It is in India, right? Whatever the way you are in Bangalore, the same way you can be in Chennai. Same thing. Use your experience. That is how you can crack. You cannot be the boss in all the subject by simple strategy. You can follow this one. The next one is a very famous picture, right? So what is this? It is cell-fate. Once it is determined, it is not going to revert back. When it is determined, if it is before determination, if you remove the cells, do you think that it will change or not? That is what we are going to see. So this is called your specialized cell, how they are differentiated. Keratinocytes. These keratinocytes will produce keratin, which is a function, protection against aberration and then desiccation. And then melanocyte, melanin, this is a pigment production, which is very good against all our UV rays. That is what they say, that pancreatic islet cells, it produce insulin, which is involved in the carbohydrate metabolism. This is how each and every cell type is differentiated. This is called differentiation, right? And we are saying that mesoderm is producing. This is what happening during gastrulation. Each and every cell will be first specified and then differentiate. Without specifying, how can it differentiate? Right? That is the process we are going to see. So the commitment, the cell fate, broadly can be classified into two methods, two mechanisms. One is specification and then determination. In the specification, there is a liable phase. It can be, so it is just like how you make your foundation for your house. This can be your living room or your bedroom. It is not determined. That is it's a outmost like a blueprint, so that we can easily relate. But once you made all your walls, once it determined, do you think that we can change it? That is determined. It is second phase, which cannot be changed at any cost. Clear? So during specification, the fate of the cell, just remember this line, the fate of the cell or a tissue is said to be more specified when it is capable of differentiating autonomously when placed in a neutral environment such as petri dish or test tube. But at this stage, the commitment is still capable of being reversed. It is not fully committed. It is like cat on the wall. It can be, may not be, maybe. That is the condition. That is called your specification. Whereas your determination, it is the stage where the cell or the tissue is said to be determined when it can be capable of differentiating autonomously even placed in another region of the embryo. Here what will happen? It should be in the neutral environment. If you keep it in any determined place, it can be reverted back. See, for example, during specification, the cells from the mesoderm, if you take it to ectoderm, it will form ectoderm organ. Once it is differentiated, even you can take any cell from any place, any layer, from ectoderm to mesoderm, it will be ectoderm product only. That is the thing, what I want to say here. So the modes of specification. So what are the different type of modes it can be specified? First, rough sketch like. It can be your autonomous specification, conditional specification or syntheal specification. During this autonomous specification, mostly in invertebrates. Okay, so specification by differential acquisition of certain cytoplasmic molecule, that is some molecule is involved in that, and then invariant cleavage produce some lineage. Blastomere fate are generally invariant. It can, because mostly invertebrate more prone for mutation. Anytime it can happen, anything. Okay, and then produce mosaic development. Cells cannot fate if the blastomere is lost. Okay, so it will be mosaic, like it can be random, anywhere it will be. Conditional, mostly it is vertebrates and few invertebrates. If the blastomere is lost, means what? The product itself will not be produced. So that is what it says. Conditional, so only the specific condition provides, persist, then only it will be determined. So that is happening in our vertebrates. Specified by interaction between cells, the cell signaling. Very good example, our part 6. The part 6 will active only in the presence of the ectoderm. Nowhere else it will be involved. If the part 6 is not there, no sensory organ. Like that. So it is totally conditional. And then variable cleavage produce no variant fate assignment. Everything will be under control. Massive cell rearrangement will take place, and then regulative development, step by step, it will be during the conditional specific. Syntheal, mostly in the inside, that is your entire homeo rule genes, pair rule gene, segment rule, that is how it will be produced. So specification, these are different patterns of specifying. Okay, it is just like even if you build a house, what you will do? Some people will go to a civil engineer and then they will have a block. Some people build it. We'll see afterwards, right? So that is how people will go. People will be with very good preparation. Some people will not be that much arranged. So this is how the autonomous on its own, whether it is other cell is induced or not, this will start doing it. Okay, but the conditional, if only A is present, then only the B will come out. Okay, so this is your different type of specification. One more time, this is exactly a line from your Gilbert. The cell fate of the cell or the tissue is specified. which is capable of differentiating autonomous on being placed in the neutral environment with respect to developmental pathway. A embryo will show a development pattern based on its type of specification. Based on the above fact, it can be said the potency of the cell is go through each and every option A, B, C, D. Remember whatever I said till now. So even you cannot use my strategy. But still you can use my strategy. You can answer even if you don't remember anything. Anuja answered first. Preeti answered second, but both of them are different options. That is, both of you guys here also, offline also, opted A, B, C, D. B, C and then A, D. Right? Yeah, A and D, she opted for B and C. Anuja, just now I explained about mosaic development in your autonomous specification, right? So this, what is your option? Benazir, what about you?
Not sure, but I think I'll go with one.
We'll go with 1. Okay.
But I'm not able to understand this question, sir. Only three options I was able to understand. The two from
So I told you, sometimes they cut and paste some line. Cut and paste the line, but we cannot get it anything. Are they asking anything different? If they use different development specification types, then I can answer. But this is your CSIR question. No, we cannot have. We have no right to ask a question.
Sir, but what if the pattern of question is pasting some paragraph, asking something else?
That's what I said. We have, but still, I'm saying that what we can, if you know the thing, so how it can be done? Definitely. So from this, what I'll do now, equal to something, the specification. I know that what is about specification, so it's going to specify. So which has to be autonomously. So which one is better, specified or determined? I'm saying that determined is more happening in a human being, whereas the specified in the primitive thing. So which is more regulated? Definitely the evolved one is going to be more regulated, right? So I can go for equal to normal fate, I can cross it. Definitely it is not going to be the same in an uncontrolled and then controlled manner. So it can be greater than normal fate in the regulative development. Definitely if it is everything regulated, everything for sure. I can keep the option B. So by having B, I am going to cross 2 and then 3. Okay?
So what does it mean? Potency of a cell greater than its normal fate in regulated development. So what this sentence means, sir?
Where?
So if we take the potency of a cell is greater than its normal fate in regulated development. So what does it mean?
See, you cannot take the exact meaning. If you see that all the 4 options are from your 3 different type of specification. Autonomous, specified and then Cynthia. Okay, so the thing is we have no other go. This is the only way. So if you keep in the regulated development, do you think that the unspecified cells will be very good in a normal way or in the regulated way? So that is the answer. So it is just literal meaning we want to take. Based on that, I can go with 1 and 4. The greater than the normal mosaic, definitely not going to happen greater than the mosaic, because mosaic itself what? Random development. So that I can go with my C. So that is how I have cracked. What happened? It will not be random. How it can be greater? Already it is confused. It's a mess. Right? So that is how it can be. Okay. So the next one is, we'll see about what happened. So generation of proximal and then distal axis. So if you see that, it is like how the mesenchyma and then different steps are going on. Let me show you the picture. Okay, it is an experiment how the apical endodermal ridge, that is AER, on the underlying mesenchyma, how the wings are produced. That is the one way. So if you remove this ARE, so ARE is nothing but what? It's an apical ectodermal ridge, so which is going to go for a production of your organ in the, especially your wings. So if you remove this one, if you remove this ARE from your forelimb, and then if you keep it, if you remove, what will happen? If you remove this ARE, what happens now? If the ARE is removed at the time of limb development, the distal limb element ceases. No any limb will be produced. So which means that I can clearly say that ARE is involved in the limb formation. So the next one, if you add extra ARE, what will happen? Person who is having 6 fingers, some people will have 3 legs. Just now I am telling apical ectodermal ridge, it's a cell. Apical meristem, like a plant, the same way they have named it. A cell which is going to produce your wing. What will happen? Extra AR is grafted in the existing limb. Supplementary structures are formed usually toward the distal end of the limb. If you see that, this is your 6th finger, extra leg, that is a problem. If you keep your egg mesoderm here, if your leg mesenchyma is directly placed beneath your wing ARE, that is from the chicken, the leg mesenchyma is kept inside the below the wing, what will happen? Your toes develop at the end. It's a leg. Do you see that? This is all your what? Toes, chicken toes, which is present in the finger. Okay. So what happens now? However, if the mesenchyma is placed farther than ARE, the hind limb mesenchyma become integrated into the wing structure. Somehow they will signal and then form. Okay. The next experiment. If the limb mesenchyma is replaced by non-limb mesenchyma beneath your ARE, what will happen? Your ARE will be totally stopped. So these are different conditions saying that what? Your ARE is involved in the formation of wings. So different experiment. Any question till now on this ARE? Which one? This is what now? Nothing but the wing is there, bird wing. They took the leg mesenchyma, stem cell. They kept it inside the wing. What happened now? The wing will be produced. Instead of your finger, you got all your leg fingers. Instead of hand finger, wings are not having any fingers. It will be like this, but it has a leg. Just imagine instead of my palm, my leg is there. That is what literally. Okay. So why? This one? What they are doing now, see here, if they keep the ARE, this one is a non, so this is a non-mesoderm, and then if you keep that limb mesenchyma is replaced by non. If it is not at all, what happens? Replaced. They are removing the entire, scrape it out, and then keep the and some stem cell which is not at all going to be the limb. So no growth is going to be there. You must have seen some persons without any hand. They will not have any sign of hand growth only, because this may be some problem. I didn't give that because that is not related to our next question. It is not at all required. If you see that ARE replaced by FG by B, so nothing but your enzyme which is artificially made so that the normal wing is coming. That is what they say, that we don't want to go for that. And then this is the question 99. Till now all of them are from development biology. Okay, now all the options. Did you see didn't feel that you are read just now? What is the Gurkamal is having question, or she answered? Very fast, that is. Very easy, right? It's a part C, 4 marks. Right? Have you used the strategy? By some other FGB. Because FGB is analogous to AER. Any question, guys? Here also, by straight, A is correct, B is correct. But we can cross this one. But don't do that one. Go for the next options and then see that definitely this is not one, this is not the one. We can go for this. Even though I don't know whether D is right or wrong. Leg mesenchyma, do you think that no leg only will be formed? That's the fourth option. It says that it will produce your proximal hind limb structure developed at the end of the. No. It has to be ARE. Mesenchyma, leg ARE should be kept. Okay. This is this option. If limb mesenchyma is replaced, the development ceases. Okay. D. Limb or leg mesenchyma is replaced by normal limb mesenchyma, non-limb mesenchyma. What happened? The ARE regresses and then limb development ceases. What is that? Leg mesenchyma is placed directly beneath the wing ARE. Proximal hind limb structure developed at the end of the limb. No. Okay. Okay, we'll go to the next one. So I think this is all about your radio immunoassay. How many of you have an idea about radio immunoassay? This is your unit number 13, methods in biology. So till now almost like 6 questions, right? In development biology, part C, not even part B. If you can crack this one, you don't want to analyze anything. Straightforward. Okay, so radio immunoassay involves the separation of a protein from a mixture using a specificity of antigen-antibody interaction, and then it will be quantitated by the radioactive element. The technique was introduced in the year 1960 by Pearson and then Yolk, and then it is actually was using assaying the insulin from the plasma. This technique is also revolutionized the entire research area, and then it is used in the blood banking, diagnosis of allergies, endocrinology. Till today, the endocrinology is in the field. This radio immunoassay is the number one. So if you have seen that, if anybody wants to go and check your thyroid, they will not do in the normal labs. They will be sent specific big labs where they will perform radio immunoassay. How they perform? If you see that, first of all, what they will do now, this is a very basic principle. Just without any confusion, just follow what I am saying. This is an antibody specific to some antigen. First of all, they label it with radioactive antigen. So this first of all, they'll label the antigen. So antibody will go and bind. So now this is called your first antibody, and then they will add the unlabeled antigen. So they want to find a new antigens. Okay. So now what they will do now, this radioactive antigen will replace with your, okay, I will show you, I will tell you with the real time. For example, I want to check the level of insulin, whether it is increased or not in the plasma. I want to check the level of insulin in the plasma. That is my idea. So for that, what I am doing now, first I am taking insulin, I am labeling it with antigen. So that is what I am doing it here, all the red color. To that I am adding an antibody which will go and bind to my radioactive labeled insulin. So I am keeping this mixture. To this I am going to add a sample in which I want to find what is the concentration of my insulin. If the insulin level is very high in that sample, what will happen? Big and antigen, unlabeled antigen present in the sample will go and bind to your antibody, so that your radioactive element will be removed from your antibody. If there is very less or no insulin in the sample, what will happen? There will not be any replacement with unlabeled, so your intensity of the radio labeled antibody at the end of the day is directly proportional to the amount of your sample present in the amount of antigen present in the sample. Understood? Any doubt? Just like in your ELISA, this is, I can say that indirect ELISA can be. Okay, so this is your basic principle, big and RIA, and then here in the RIA, what is that? Advancement method of ELISA is RIA. In ELISA, actually, like if you see that RIA, we don't have any idea about fluorescent molecule first. We have used only radioactive element that time. So the advancement of RIA is ELISA. That is, if you use this radioactive element, we need, see, for example, if I want to check the data, I want the Gregor counter, scintillation counter, because it has to be gamma radiation, it will be cancerous. So many problems are there. So that mostly till now, people are using for thyroxine, thyroid thing. And then next one is the application of this RIA. Okay, that is all about your RIA. So that directly the question is like this. It is 136th question. You got the principle right, what I said about RIA, everyone. It's very simple. Real scientist analysis question, this is. What is the question is all about? You performed RIA, that means what? You must have labeled your antigen first, and then you must have added your antibody, and after that you must have added the sample. At the end of the day, you are not able to see any signal. What is the explanation you can give for this? Online students? Yeah, the antibody. That is titanium is a radioactive element. Tritium, sorry. too much of time for this. Okay, I don't have any idea. Okay, radioactive hormone was insufficient. The sample is insufficient, definitely not. That if it is less, there is a big difference between insufficient and then sufficient. Means, for example, in the case of, if this is an experiment, nobody will start an experiment with insufficient sample. Definitely A I will cross. That is not at all. The following examples are proposed. Hypothetical conditions. C is for definite. Hypothetical conditions. See, people what they'll tell. Tomorrow will be raining, so that I will not come today. Such answers. It's a lame excuse. That is what I'm asking. So I will cross A, definitely. Because see, the thing is, like, if this is your condition, your PA is asking, okay, sample is not insufficient, he'll kill you. Because he is the one who will give the sample. Definitely it will be sufficient. So definitely A is, you cannot give any excuse. And then B, the radioactive tag to the hormone completely disassociated during storage. It can happen. All of them, because if your storage is not proper, if your buffer is not proper, this disassociation can happen. Because when you wash, because if there is any trace amount of radioactive, it will light up. The signal will be light. That is the beauty of radioactive element. If there is no any radioactive element, then only the problem. If it is very, very less, for example, in the place of 7 microcurie, you want to use, you have only 3 microcurie, definitely it will work out. The trace amount of radioactive element also will work out. So that could be the very good excuse. Electricity gone, so the fridge is switched off, so the temperature increased, disassociated. Yes, that's a very good excuse. The antibody was not added to the cocktail. That can also be done. Okay, that is also very, I forgot. Because always people will be excitement. In the case of second solution, they'll go for the 5th solution. They'll wash everything, they'll throw it off. And then, what happened? See, they have given a situation in that radioactive glucocorticoid was added to RA cocktail. Okay, so they didn't see any counts. So what could be the possible thing? Excuses. So the C can also be the options are my option. Which one? So this is the radioactive antigen. No, it is not our sample. It is already, because for example, if you are going for insulin, it will also be the same insulin type of antigen. It will be radiolabel. They will go for your antibody. To that sample, they will add a free antigen, that is which you want to assess. So that what will happen, there will be a fighting between the binding of this radioactive labeled antigen with your sample which you want to detect. So it will go on bind. And then if you wash it, everything, all your unlabeled free antigen and then radiolabeled antigen will be removed off. At the end, you will have only the sample which is with your thing. So this will light up very little, so the signal will be very, very less. But in the free solution, there will be activity will be very high, which means that it has free more radioactive element when compared to the sample. Okay, I'll tell you one more time. Okay, wait. I'll explain. It's very important. If you have, because if you are asking question means if you understood the basic, that is, it is all about your competitive binding. Okay. First of all, they will take a protein A, which can bind to the antibody. So the binding will be always weak binding. Okay. So the protein A is labeled. So I was given a solution which contains antigen B. Okay, I want to find what is the concentration of this antigen B. This antibody which I going to use is having more affinity to antigen B when compared to antigen A. Got it? So I'm mixing this antigen radio labeled with antibody. I'm keeping it for some time. So what happens? All my radioactive element will go and bind to the antibody. The leftover I'm washing it off. So I have only antibody which is labeled with antigen. To this I am adding the solution which contains my antigen B, which is blue in color. Okay. Basically the antigen B will have more affinity towards the antibody when compared to antigen A. So now there will be a tug of war between antigen A and B. It's my place, remove, move. And then environment will be given more favorable for binding of antigen B. So in that there will be competitive binding. So after that what will happen? Some of the antibodies which is labeled will be removed and then unlabeled antigen will go and bind. Clear? So in that case you will wash the solution so that it will have some free antigen which is labeled and then as well as unlabeled. In that if you see your radioactive scintillation count, I think you hope you must have seen that it will make a sound. Okay, based on the sound, and then if you perform your autoradiograph, based on that you will get a signal.
The third option like antibody not added, that is the primary antibody or secondary antibody?
There is only one antibody here.
And this secondary antibody we can add in.
The secondary antibody is only to remove your bound one. Okay. Okay, the option is your C. Next one. So this is a primary metabolite. So we know that mostly it will be produced in the exponential growth, and then secondary metabolite mostly produced in your stationary growth, and then stationary metabolites are not at all responsible for required for the growth, and then mostly depends upon the growth condition. More number of mycelia, for example any antibiotic penicillin, if the penicillin mycelium produces more, the product will be very high. So this is our, and then to be more specific, we have seen this one. In the exponential phase, the primary metabolism will be produced exactly at tropophase. In the stationary phase, it will produce exactly at idiophase. Clear? This and all we have seen that. So this is your question number 37. Can you hear me now, everyone? Guys, can you hear me? Okay. So, which graph exactly shows your production of your Streptomyces species antibiotic production? The answer is 2. If you see that there is no any much difference between graph 1 and 2, and then even for 3, but if you see that in this one the nutrients are depleting, but it is not like that. Actually it will be there for some time and then it will be depleting. That depletion will increase your production of your product. And then 3, if you see that the biomass, and then it is not always high, because antibiotic will never be produced in your exponential phase. It will be produced only in the stationary phase. The option is 2. So next one is your transgenic crop. We have seen the transgenic animal and the transgenic plant, and then which is involved in the crop improvement. If you see that the crop improvement refers to the genetic alteration of the plant only to satisfy our human needs, and then one of the most important technique which is used by your crop improvement was gene silencing. Is nothing but what? Anti-RNA silencing. Okay, first it was developed by the scientist in the potato virus resistant plant, and then after that it was seen in the so many plants like gold bean mosaic virus, banana brat mosaic virus. Again, so many virus they have use this gene silencing. And then the next one is, in addition, even the plants can be used to modify double-stranded RNA that silences essential gene in the insect pest as well as in the parasitic nematodes. So gene silencing, to be more specific, it can be defined as it is switching off a gene by a mechanism other than genetic modification. So that is a gene which can be expressed, or under normal circumstances it can be switched off. So that is what the antisense RNA. It can be expressed, it can go on bind. Once a double-stranded RNA is produced, no more translation of your protein will take place. This is all about your gene silencing. With this background, online students, can you hear me, right? Okay. Sadesh, what is the answer? Benazir, what is the confusion here? Third and fourth. If it is co-suppressed, which means that it is suppressed by some other gene. Production of A will suppress B. If the A is important protein, definitely the plant will not grow. What is co-suppression? What is suppression? Not expressed. Co-suppression means with the help of some other gene it is suppressed. If with the help of some other gene it is suppressed, means what? So definitely it is not good, because it's a new gene which will express. There is no any chance for that. Right? Definitely not a gene knockout, because it's a transgenic. We are adding some gene. So suppression of effect of transgenic, definitely not, because we are going to see for a expressive plasmid which will go on express, it will go on insert, because it happened in 30 out of 30, 20 to show the expression, but only 8 is not having. So most probably problem is, yeah, that is what, there is no, yeah, some of the, see, that is what we don't want, but it's happening. One of the most possible mechanism by which the expression of the gene can be done. Okay. The answer is your 3.
It means like gene silencing will not lead to complete silence of the gene. It will somehow there will be there.
If we are here, we are not doing any gene silencing. We are producing transgenic. In that, I am seeing low level of expression. So most probable answer is it can be because of gene silencing. The plant somehow evolved producing some mRNA which is related, partially related to that transgene. Yeah. Next one. Stem cells. So already we have seen this slide. If you see that, up to 5 to 7 days of your embryo will be totipotent. After that, it will be, after 6 weeks, it will be pluripotent. That means that fate is determined. Understood? Cell fate is determined. This is all about your stem. Already we have seen that. Just to refresh it. The next one. Others can hear me? Which one? Yes. Embryonic stem cell. That is a blastocyst. So if you see that, this is how they will collect your embryonic stem cell. The human, it was happened in the 1998, and then mouse it was 1981. This is your human blastocyst. This is called your trophoblast. And then inner mass cell they will collect for your transgenic plant or transgenic animal. And then this is all your question. So the difference between the totipotent and then pluripotent is totipotent, it is not fate is determined, whereas pluripotent is fate is determined. That is the pluripotent stem cell, for example, the cell which is there in the bone marrow, which is always for your blood cell. Okay. So here the option is what they given. A person liver is damaged. How you will correct him? What is the possible way you can give? Third one, embryonic stem cell is your totipotent, which is not fate is determined. Adult liver cell from healthy donor, there will be a problem with your immunology. Yes. Induced pluripotent stem cells. If the option is 2 is not there, then we can go with your 3. Because instead of injecting the stem cell, we can induce it. That induced word is the right one there. Okay. So the second one is your Nobel Prize winning somatic cell to stem cells. Inject. What is that? If you are talking about gene, if you are not specific, I cannot answer. There is a damage in the wall. Okay, the wall is wooden wall. What you will do? Answer this. Answer this. It is not. No, I cannot. Just now I told you why you want them. What is pluripotent? What is totipotent? Okay. If you want to go for immediate result, differentiated. So first question, if you collect the cells from adult, the problem will be your MHC. The second one, you don't have any idea, leave it. The third one, injecting embryonic stem cell, yes, it can be, and then injecting cord blood cells directly into the liver, it cannot work out, because cord liver cells also is nothing but your embryonic stem cells will be just not thing. But if you read that transforming skin cell, that is nothing but your stem cells. If you are taking the same embryonic stem cell and then you are from the same patient, that is very important, from the same patient, so that you can avoid all your immunological reactions. Number one. Next, that stem cell was induced into the pluripotent stem cell, that stem cell can be producing your liver cell.
Any cell can be taken to induce into differentiated cells. In cell state, any cell can be taken.
This is an hypothetical situation. If it is there, that is what I said, that 2012 a person got Nobel Prize only for this, to convert your somatic cell into stem cell. That can be, this is a future.
So stem cells already differentiated cells.
Yeah, that is what I'm saying. Now we have an option. Which of the following strategies would be the safest, which you will opt for? None of them are available right now. Nobody will inject the stem cell directly into liver cell. It will not happen anything. Okay. So with the information what we have, we can accept any organ from the next person only if we are immunologically compatible.
And how can differentiated cell can be converted into the same?
That is a Nobel Prize. We have the protocol, we have the procedure.
No, they are reverting it back to the
repriming to the pluripotent stem cell. That is the 2012 Stem Cell Nobel Prize for Medicine. Not how we have the protocol. It was imaginatory. We have soon, in another 10 years, the option B will be our treatment. Injecting cord blood is nothing but your placenta that will have a large amount of your stem cells. That is, it will create a problem. And then if they didn't specify also, even if you don't have any idea about that, from the patient, that is the clue. We can preserve. Yes, that's possible, but it's very, very costly. Almost for 10 years they are asking 40,000 rupees. Next one. I think we will stop here. I think we'll stop here. We'll start the class from next second one. Straightforward. Option is 2. Okay, I'll explain why it is so the next class.